Our Dr. Shephard and Dr. Phillips will get to that question as the webinar starts. So my clock says that it's about time to go, and we got 15 folks here, so welcome everyone. I think I'll just throw it over to you now, Dr. Shephard. Is that okay?
That's fine. Can you hear me? Can you hear me?
Yeah, I hear you fine.
Well, just welcome to everyone. It's the evening in London. I'm not sure where you're all coming from, but thank you very much for asking us to present this webinar session. And as the name says on the slide, my name is Elizabeth Shephard, and my collaborator Ian Phillips is also present and sitting next to me, and we'll be happy to try and answer your questions if any arise. And what I've done tonight is I've entitled our presentation FMO3, Bugs, Genes and Drugs. I have the next slide, please. So the protein FMO3 is the one that is responsible when mutated for the condition trimethylaminuria. Now FMO3 has this rather long name called flavin-containing monooxygenase 3. And the name really says everything about the enzyme. The flavin is just a small molecule that has to be attached to the protein for the protein to have its function. And actually that gives the protein a very pretty color, a bright yellow color when you isolate it from the cell. And the monooxygenase part of the name refers to the fact that the enzyme adds an atom of oxygen onto the chemicals that are its substrates. So mutations are changes in the base sequence of DNA, so a change in an A, a T, a C, or a G. And sometimes these changes can have unfortunate consequences because they can alter the ability of a protein to do its job properly. And such changes can lower the amount of protein produced, or they can eliminate the activity of the FMO3 protein. And this is when we have the condition trimethylaminuria, when we have a compromised amount or activity of the FMO3 protein. Now, in the first part of the talk, I'm going to talk about trimethylaminuria, but FMO3 does much more than that, and it also has a role as what we call a drug metabolizing enzyme. And the second part of my talk, I'll talk a little bit about how it plays a role in drug metabolism. Could I have the next slide, please? So what I'd like to do here is to introduce you to the concept of bugs and the gut microbiome. If we simply look at the number of cells that make up the human body, we have a lot of cells, and it's about 10 to the 13 cells. But our intestines contain at least 10 to the 14 bacterial cells. We don't really know how many bacterial cells there are. We just know there are 10 times more bacterial cells in our body than actually make up the human body. So if you think about it, actually we're more bacteria than human. But the mass of the bacterial cells is smaller than our mass. Now, it's quite interesting. As we've sequenced the human genome, we found that we only have about 30,000 genes that code for proteins. And a very simple worm has 14,000 genes. That's not very much different then of what takes it to make a worm or a human. And so people are starting to think of how we can actually do all the reactions and make use of all the nutrients with such a limited number of genes. And there's now a lot of increasing evidence that the gut microbiome, all of the reactions that the bacteria carry out in our gut, is providing us with a second genome, providing us with key nutrients and key reactions that we ourselves cannot carry out. So the gut microbiome is extremely important to us and is recognized as a key factor in health and disease. Could I have the next slide, please? So it's really the bugs in the gut that cause the problem of trimethylaminuria. When we eat food, and a lot of foodstuffs contain a chemical called choline, some of the bacteria in our guts change the choline. They chop off a small piece of the choline molecule to create a small molecule which is called trimethylamine, or TMA. Now, trimethylamine, it's a very small molecule. It has a group that we call a methyl group. That's 1 carbon, 3 hydrogens. That's the methyl group. We say CH3. You might have seen it written like that. And we have 3 of those. So it's trimethyl. And they are attached to a single nitrogen atom. And that's what makes trimethylamine. And it's trimethylamine that actually is the compound that causes the ammoniacal odor. Now, why do the bacteria produce this chemical? They produce it because there's a certain class of bacteria that actually use trimethylamine as a nutrient. And the more trimethylamine they produce, the more they increase the production of the enzyme that produces more trimethylamine. And so it's kind of like a vicious cycle in terms of trimethylamine production. But trimethylamine is rapidly absorbed into our bodies. And in the liver, we have an enzyme, the enzyme FMO3, that converts trimethylamine to trimethylamine N oxide. That means that the enzyme adds an oxygen onto the nitrogen of trimethylamine. And so we call that the N oxide. And so we convert a chemical with an ammoniacal odor into one that does not have this odor. Now, if something goes wrong with the FMO3 protein, then this reaction is not carried out effectively, and that's what then gives rise to the condition known as trimethylaminuria. Could I have the next slide, please? Now, this is a rather complicated looking slide, but it has a simple take-home message, and that is that if you just look at the bar, that's the blue and white bar, you'll see a number 1 at the left-hand side and a number 532 at the other end. This just represents that the FMO3 protein is made up of 532 amino acids. If you look at if I move the cursor, can you see me using the cursor? The vertical lines represent positions in the FMO3 protein that have been changed because of mutations in the FMO3 gene. And you can see that there are a lot of mutations that can occur. Some are specific to an individual family, and some are found in quite a lot of people that have trimethylaminuria. If we just concentrate on the position, if you can see the position P153L, it's in the P is in orange and the 153 is in blue. What that means is that at amino acid 153, an amino acid called proline, P, has been changed to an amino acid leucine called L. And that one change has caused a protein that had a function when the proline was there, but when it was changed to a leucine, now the protein has no enzymatic activity or very little enzymatic activity. And that's true for all of it. As you go along this bar, you can see different positions. One other one I'd like to just point out is E305X, if you just run your eye along the bar. X means stop. It means a change in the DNA that says stop protein synthesis. So in a person that has that mutation, they make a protein that's only 305 amino acids long. And of course, that's a protein that is too short and cannot carry out its function. So mutations can change the activity of the protein. They can shorten the protein. And really, the severity of the disorder will be dictated by the mutation. So some mutations will have a less severe effect than others do. Could I have the next slide, please? So just to tell you how we name amino acids, because it can be quite confusing when you look at some of the literature. So amino acids, there are 20 that make up proteins. I'm just showing 6 of them here. And you'll see on the left-hand column the full names of 6 of these: alanine, glycine, glutamic acid, leucine, lysine, and proline. Now, in the olden days, when we just started to learn about amino acids and that, it was fine to write out the full names. But as we started to be able to sequence proteins and determine the amino acid sequence, the order of the amino acids in a protein, it got a little bit tiresome to write out the full name the whole time. And so we came up with a 3-letter code. That's very simple. It's just the first 3 letters of the name of the amino acid. So alanine is ala, glycine is gly. However, when we were able to sequence DNA, and now we've got the whole genome, the human genome and many other genomes, it just takes up too much space to use the 3-letter code. And so now we use a 1-letter code. And this just makes it very easy to represent a protein sequence, an amino acid sequence. There is a logic to the 1-letter code. Alanine is A, glycine is G. Glutamic acid also starts with a G, but G's already been taken by glycine, and so we use the next letter that's closest to the G in the alphabet that hasn't already been taken, and that's E. There's no amino acid that starts with E. Leucine is L, lysine is K, because the L has been taken by leucine, and so we use the next letter in the alphabet, which is K, and proline is P. And as I said, X is stop. So if you do read the 1-letter code, you can usually guess at which amino acid it is, because most of them start with the first letter of that amino acid, and they're just the few odd ones where the letter is the same, and so we have to have another 1-letter code. Could I have the next slide, please? So mutations in DNA that affect the activity of the FMO3 protein and which result in trimethylaminuria, we call that form of the disorder primary TMAU. It has a genetic basis. It's a direct correlation between the genetic information and the protein and its inability to do its job properly. But how do we actually do this in the laboratory? How do we know that these particular changes cause a defective protein? Because we all have changes in our DNA, and many of them don't influence us at all. But occasionally, of course, they do. So as a scientist, what we have to do is we take the DNA sequence of a person that does not have trimethylaminuria, and then we see a change in a person that does, and we artificially create that mutation in the laboratory. And then we take that DNA sequence, and from this, we use this to produce the protein. And then we test whether the mutant protein has activity or not. If it has no activity, then we know that the mutation is the one that is responsible for trimethylaminuria. So if we look at this little graph here, on the left-hand axis, we've got enzyme activity, and on the bottom axis, we've got time in minutes. And we're looking here at the ability of 2 proteins to convert trimethylamine to trimethylamine N oxide. Now, the protein that has a proline at 153, you can see a very nice straight orange line. So this enzyme is very good at converting trimethylamine to trimethylamine N oxide. However, when we change that proline at 153 to a leucine at 153, you can see a dramatic change. The enzyme has practically no activity whatsoever. And so this is absolute proof that if a proline at 153 is changed to a leucine at 153, then that is the mutation that is correlated with trimethylaminuria, that is causing the disorder trimethylaminuria in a patient that has that mutation. And that's how we do it for each of the mutations. You can't just guess at the change, you have to prove that the change changes the enzyme activity. Could I have the next slide, please? Now, we can prove that changes in DNA are responsible for the disorder, But most people would have a urine analysis diagnosis for TMAU before they would have a genetic diagnosis. And the urine analysis, as many of you will know, measures the concentrations of trimethylamiine and the amount of trimethylamiine that has been converted to trimethylamine N oxide. And so the less trimethylamine that's been converted, the more severe the disorder. Now the results are usually given as a percentage, and we express this as the amount of trimethylamine N oxide that is excreted in the urine divided by the amount of trimethylamine that's excreted plus the amount of trimethylamine N oxide, and multiply that by 100. If you're unaffected, then your percentage will be 90 to 100%. But if you have a mutation that moderately influences the activity of the enzyme, then the percentage will drop between 40 to 90%. And if you have a protein that really can't do its job very well, then the percentage will be less than 40%. And so this is a good categorization of the severity of the disorder. And then perhaps if people have this test, then they may wish to have their DNA sequenced to see which mutation they carry. Could I have the next slide, please? So that's primary TMAU, a direct correlation between your genotype and the FMO3 protein. But there is another form of trimethylaminuria. We call this secondary trimethylaminuria. And these forms are less well understood. They may well have and do have a genetic component, but they also have an environmental component. So these are much more difficult to investigate. There isn't just a clear-cut genetic effect. The environment comes into this, just like it does in disorders for diabetes or other disorders. So there's a form of trimethylaminuria we call acquired trimethylaminuria, and this occurs in some patients that have viral hepatitis. And what happens here is the patient recovers from the viral infection, but has acquired trimethylaminuria. This is rare. Most people will recover from viral hepatitis and won't acquire trimethylaminuria, but some people do. And we do not know why their FMO3 protein is compromised by the viral infection. A common cause of secondary trimethylaminuria is bacterial overgrowth in the intestine. This means that the spectrum of the bacteria in the gut has changed. So instead of being sort of a normal balanced kind of population, there'll be an overgrowth of particular bacterial class. And this will then result in a large production of trimethylamine. Certain disease states of the liver and kidney are also known to produce the symptoms of trimethylaminuria. And then we have a class of trimethylaminuria that we call transient trimethylaminuria. Some children present with the symptoms of trimethylaminuria, but they grow out of the symptoms. Now this undoubtedly does have a genetic component, but the gene is really quite interesting. The gene is only activated at birth, so it's not active in the fetus. This is a common thing in our development. Certain genes are only switched on at birth, and the FMO3 gene is one of these. Gets switched on in the liver at birth. And we think that these children present early on with these symptoms but grow out of them because their gene is delayed in becoming active. But as they grow and become older, this gene becomes activated and then starts to produce a sufficient amount of protein to cope with the trimethylamine. Usually the children may also have, one of their genes may be delayed and one of their other genes might carry a mutation or a polymorphism that reduces their other protein a little bit. As you know, we have two genes, one from your mother, one from your father. And in terms of the liver, until we're about 7 years old, our liver really isn't a proper liver. The liver of a child is very different to the liver of an older person. We know that menstruation can be present or increase the symptoms of trimethylaminuria. This is probably because FMO3, if there's any activity there, is inhibited by hormones. And when that hormonal surge has changed, then the condition kind of wanes, but will recur through the monthly cycle. And then we have precursor overload. This is when a person eats an awful lot of precursors for trimethylamine. So a diet very rich in choline. Perhaps they've eaten a lot of soya. Soya is very rich in choline, or they've been given some tablets that contain choline. But this is simply the body is just overwhelmed by the amount of trimethylamine that's produced. And again, that condition will subside when the precursor overload has been reduced. Could I have the next slide, please? Now, there are other factors that increase. I see one person has raised their hand. Would you like me to answer the question now? So there are other factors that increase trimethylamine in the urine. These are urinary tract infections, bacterial vaginosis, cervical cancer, and some other conditions. And these can be misdiagnosed at first glance as trimethylaminuria, but a urine test will show that these are not trimethylaminuria. They're just an increase in trimethylamine in the urine. And the difference is that if the trimethylamine and trimethylamine N oxide ratio is calculated, it's normal in these individuals. So they have a normal ratio, it's just that they are, because of this particular condition, are at that time producing a lot of trimethylamine. Next slide, please. So these are two information links. I mean, there are others out there, but one is the Gene Reviews, which is an NIH resource on trimethylaminuria, and then a European resource, which is provided by EURORDIS, which is a clinical utility gene card for trimethylaminuria. If you go to the EURORDIS site, you have to run down the page until you come to the T's, click on trimethylaminuria, and you can download the PDF. Could I have the next slide, please? So the next part of the talk I'm going to devote to other actions of FMO3. Now, I said that FMO3 is a drug metabolizing enzyme. And in fact, that's why we were first interested in it, because we have worked for a number of years on drug metabolizing enzymes. So FMO3 does play a key role in trimethylaminuria, but it can metabolize chemicals other than trimethylamine. And a group of very important chemicals that are metabolized by FMO3 are therapeutic drugs. Now, drugs like foodstuffs are foreign to the body, and they have to be removed. And we have evolved very sophisticated defense mechanisms to clear foreign chemicals from the body, and this is a process called detoxification. So you might think, well, why do we need this system? We need it because when we eat foodstuffs or we take a drug, we take them in a very high concentration. And the body responds by trying to rid itself of this excess chemical. And that's the process of detoxification. And detoxification can occur in many organs in the body. This includes the gut, lungs, skin, and kidney, for example. But the real powerhouse for detoxification is the liver. And here, as you saw with trimethylamine, many chemicals undergo changes that allow them to be removed from the body in the urine, the bile, or the feces. And therapeutic drugs are foreign chemicals, but they've only been around for a short time, a lot shorter than humans have been on planet Earth. And we're just very fortunate that we have evolved enzymes that can clear chemicals from our body and from food like trimethylamiine, because they allow us to benefit from therapeutic drug therapy. If we didn't have enzymes like FMO3, we wouldn't be able to make such a wonderful use of therapeutic drugs. So have the next slide, please. So this is a brief pictorial example of detoxification. And what we have here, this ground circle represents a liver cell, and the little pink triangle is here, in this case, a drug. Now, many drugs are what we call hydrophobic. They like sort of an oily environment, which means that they can pass into the membranes of our cell. Our cells are surrounded by a membrane that is very sort of, you can think of it as an oil barrier. The drug can pass through this into our cells. And of course, if we didn't have anything in our cells to kind of get rid of this excess chemical, then the cell would just curl up and die. But fortunately, we have enzymes like FMO3 who will add an oxygen onto a drug. And when that happens to the chemical of the drug can then be start on its journey out of the body. And often with, in the case of FMO substrates, they'll be excreted through the urine. So in this example, I've got the triangle with a little oxygen attached. If there's a nitrogen on the drug and the oxygen attaches to the nitrogen, we call it an N oxide. But many drugs have a sulfur, and if the oxygen attaches to the sulfur, we call that the S oxide. So the drug I'm showing as a drug, but it can be another foreign chemical, and of course I've already mentioned trimethylamiine. Could I have the next slide, please? So I wanted to introduce you to the concept of pharmacogenetics. This is a specialized area of genetics, and it's a study of how our genes influence the way we handle a drug. You might have heard the term personalized medicine, this thought that we might be able to tailor our therapeutic drug therapies to our genetic makeup. Now, when we take a drug, if we swallow a pill, that drug has to be absorbed into the body. It has to be distributed throughout the body, and it has to be cleared from the body. The excess has to be cleared from the body. And there are lots of proteins that are involved in these different processes, but clearance is one in which the FMO proteins are involved. And so changes in your FMO proteins could influence the way in which you clear a drug. Can I have the next slide, please? So why does drug metabolism and clearance of the drug matter? Well, it matters really a great deal. So there are two panels here, and if you first look at the one on the left-hand side, you'll see a graph that on the one axis we have plasma concentration, and on the bottom axis we see little pink arrows. And the little pink arrow refers to, it's sort of representing when you swallow a pill. So it's very common for a doctor to prescribe a tablet 3 times a day, morning, lunchtime, and dinner. And this is because you take a drug, the plasma concentration increases, so the drug reaches its concentration where it will be effective, and then the body starts to clear this drug. So that's where the FMOs might come in. You take another, so the concentration drops. You then take a tablet at lunchtime, the plasma concentration increases, and then we have clearance and the concentration drops, and so on. So each time you take a dose, the drug increases in the plasma, does its job, the excess is cleared from the body, and that's what we would want to happen. But if you look at the right-hand side panel, again we have plasma concentration. Take the drug, the first pink arrow, and the plasma concentration rises. But here someone has a genetic change in a gene that's involved in clearance, so that the clearance is not very good and the plasma concentration stays high. They take a second dose and the plasma concentration increases, and then again there is little clearance. And a third dose, the plasma concentration increases further. And so you can see that in this particular case, where there is a defective clearance mechanism, the plasma concentration of the drug keeps rising. And this, of course, can have very dangerous consequences, because in effect the person will be taking an overdose, and inadvertently taking an overdose of a tablet because they cannot clear the drug effectively. And this is when we have the potential for adverse drug reactions, which are a real problem in the Western world and cost health services a great deal of money, and more people die from adverse drug effects than from road accidents in both the UK and in the US. So you can see it is a serious problem, and one which means we should understand how genetics affects, for instance, drug metabolism. Could I have the next slide, please? So this is just a short table of examples of FMO3 drug substrates. And I've just chosen some whose names you might be familiar with. So lidocaine, which is an anesthetic, benzidamine. There's several antipsychotics, chlorpromazine, clozapine, fluphenazine, olanzapine, pyrazine, nicotine, and tamoxifen, the anti-estrogen. Now, one thing I don't want you to do is to worry about taking these drugs if you have an FMO3 genetic change, because the studies are limited, and as I'll show you in a minute, drug metabolism is a lot more complex than just one protein, one substrate. We do know that patients who have trimethylaminuria do have impaired metabolism of benzidamine. But for these other drugs, most of it's been done in the laboratory. We take some FMO3 protein, we change the sequence, we mutate it, and we look at the effect. And we can say, yes, this is an FMO3 substrate. Yes, this mutation influences the metabolism of that drug. That doesn't necessarily mean that this always happens in the whole body situation. This is much more complex. Could I have the next slide, please? Now, before I talk to you about a little bit more about FMOs and drug metabolism, I was asked to mention cytochrome P450 monooxygenases, or CYPs. There's another favorite family of proteins that we've worked on. They're a very important family of proteins because, like the FMO proteins, they also help to metabolize drugs. And you can see they are also monooxygenases, so they also add an atom of oxygen onto a substrate, and they're called cytochrome P450. That name is, a cytochrome is a colored protein, and the cytochromes actually are a beautiful red color when you isolate them from the cell, and the P450 refers to the fact that if you were to measure the wavelength at which that pigment has the most intense absorption, it's at 450 nanometers. Now, we have lots of different CYP genes, and I actually don't know how many CYP genes we have in the human genome, but it is really a lot, and all organisms have them. And so scientists had to come up with a way in which we could talk to each other about different CYPs and know which protein we were actually talking about. And so they looked at all of the proteins and their amino acid sequences, and then they sorted them into families according to the amino acid sequence. So we have the CYP1 family, and it has a number of members, and they're called CYP1A1, CYP1A2, CYP1B1, and there's a few more. But these proteins all have similar amino acid sequences. They are different, but they have similarities. And their amino acid sequences are more similar to each other than they are to, for example, the family called CYP2, where the members of the CYP2 family also are more similar to each other than they are to members of the CYP1 family or the CYP3 family. And two very important members of the CYP2 family are CYP2C19 and CYP2D6. And then we have the CYP3 family, and a very important member here is the CYP3A4 protein. And there's a CYP3A5 and there's a CYP3A7, and so it goes on. And so you can see how complex this family is. But many prescription drugs are metabolized by CYP2C19, CYP2D6, and or CYP3A4. In fact, about 50% of prescription drugs are metabolized by CYP3A4. And if you open a packet of tablets and you take out the little leaflet, you will often see that these days that it says this drug is a substrate of CYP2C19 or CYP2D6. And I think that's more prevalent in the United States than perhaps in European countries, although it is changing in Europe as well. Could I have the next slide, please? So I've mentioned CYPs and I mentioned FMOs, and drug metabolism is kind of a concerted action between different proteins. So drugs can be often metabolized by more than one CYP and or an FMO protein. And it might be that each of these enzymes or proteins might produce a different drug metabolite. So you have this, you start with the same drug, but you end up with different metabolites depending on what a CYP or an FMO has done to that particular drug. Or in some cases, you can have different CYPs and FMOs, and they all produce the same metabolite. It really is dependent on the drug. Could I have the next slide, please? So in this slide, I'm just showing you a simple example of how a drug can be metabolized by an FMO and by several CYPs. The drug is an antidepressant imipramine, and it's metabolized by FMO1, not FMO3, but I've chosen this example just because it's a simple and quite clean example to explain the process to you. So imipramine is converted by FMO1 to imipramine N oxide. So imipramine has a nitrogen onto which FMO1 puts an oxygen, and then imipramine N oxide can be cleared through the kidneys. Imipramine is also converted to a chemical called desipramine by CYP1A2, CYP2D6, and CYP3A4. And I've just put here metabolites, and in fact there are about 12 metabolites that are produced by the actions of the CYPs. So quite complicated, but there's a single product of an FMO reaction, which is imipramine to imipramine N oxide. Now, this also demonstrates something else about drug metabolism, in that imipramine has antidepressant activity. When it's converted to desipramine by the cytochrome P450, desipramine also has antidepressant activity. And so the body is helping to make in what we think is a more potent antidepressant from the imipramine. Could I have the next slide, please? So the story gets even more complicated when we think about FMOs and their drug metabolism and cytochromes P450, and we can have something that we call drug recycling. In technical terms, we call this retroreduction. So here we have imipramine going to desipramine through the action of cytochromes P450, and imipramine going to imipramine N oxide by the action of FMO. But the body always does curious things, which always complicates our things. There are several enzymes that, when imipramine N oxide has been produced, turn around and take the oxygen off the imipramine and produce imipramine. So take the oxygen off imipramine N oxide to produce imipramine. You can see the arrows are going both ways, imipramine to imipramine N oxide and imipramine N oxide back to imipramine. And then imipramine more imipramine going into desipramine. And so we have this drug recycling where you take a dose, but actually your body is kind of increasing that dose. Now, this doesn't happen in every tissue. This reaction actually is happening particularly in the brain, where we get this recycling. It's quite potent. It does happen in other places in the body as well. Could I have the next slide, please? So we talked about FMO3 and about CYPs and about how genetic polymorphisms can influence drug response. But why I said you shouldn't necessarily worry if you have an aberrant FMO3 protein is because maybe you've got an aberrant FMO3 protein, but if you've got a really good P450 protein, then maybe the drug's going to be cleared by a different pathway. So we have, in this example, I've called it multi-pathway drug metabolism. So here we have a drug that 80% of the drug is cleared by pathway A and 20% by pathway B. So if pathway A is functioning, that's absolutely fine, and it doesn't matter if pathway B doesn't work so well. So it's kind of a, it's an additive effect. It's the drug will go down the pathway that it can, and if it's a good pathway, it doesn't matter if another pathway is not working quite so well. If pathway A is impaired, however, and you push the drug through another pathway, you might get a different effect. And so your genetics is really complicated in terms of drug metabolism, because it really isn't just about your FMOs, it's about your CYPs, it's about other proteins that all work together to help you to absorb, distribute, and clear drugs. Could I have the next slide, please? So this slide brings me back to the title, which is Bugs, Genes, and Drugs. And it's a really nice example about how all of this works together and how the gut bacteria actually, again, are beneficial to us. Now, Sulindac is a drug that is given for the treatment of colon polyps. But Sulindac is what we call a prodrug. It has no activity as a drug whatsoever. But when we take this drug, the bacteria in our gut convert Sulindac to Sulindac sulfide. And that is the active drug. So it's the bacteria in the gut that convert this chemical into the active drug. And then the FMO3 protein then adds an oxygen onto the Sulindac sulfide to produce Sulindac sulfoxide, and that's the inactive compound. So the FMO3 is helping to clear the drug from the system. What's interesting about this example is that a group of patients was studied, and a number of them actually responded very well to the treatment, and the colon polyps regressed. And they looked at their genotype, and they found out that these patients actually had a genotype which gave them an FMO3 protein that didn't work so well. So the protein is shown above here, 532 amino acids of FMO3. And at position 158, we have an E going to a K and an E308 going to a G. So in the patients that have a lower activity of FMO3, they will have a lysine, a K at 158, but they'll also on that same protein have a G at 308. And those two changes together make a protein that is a bit less active than a protein that's got an E at 158 and an E at 308. And so in this case, it was actually advantageous to have a protein with a lower activity. And we think that the reason that the patient responds better if they have a lower FMO3 activity is that the Sulindac sulfide will stay around longer in the gut, and the FMO3 won't be able to clear the drug as effectively. And so the drug is there for a longer time, and for whatever reason, the colon polyps then regress. So it's just an example of having an FMO3 protein with a lower activity isn't always a bad thing. Sometimes it is advantageous. Could I have the next slide, please? Okay, well, I just thought I'd end by giving you an example of some non-drug FMO3 substrates, because really we talk about FMO3 as a drug metabolizing enzyme, but really what we mean is it's an enzyme that metabolizes foreign chemicals, so things that are foreign to the body. And drugs just happen to be one of those classes, as do foodstuffs or components of foodstuffs. So there's some long names here. Well, we needn't worry about the names, but certain environmental sulfides, and I apologize, I've mixed the English spelling and the American spelling here, but pH in British English and an F in American English, same thing. So certain environmental sulfides are substrates for FMO3, the pesticides, aldecarb, phorate, and fenthion, fuel additive. Phonosylcysteine is a modified amino acid. Seleno-L-methionine is a methionine analog.
Sorry about that. Oops. Sorry, I should get this going again in a second. Hold on 1 second. Sorry. Good. Okay, we're back. Sorry about that.
Sorry, when did we lose the sound?
We lost the sound just about 20 seconds ago for about 10 seconds.
Okay. Well, I'll go to the bottom of the table. As I said, we have we have the gut bacteria. They produce numerous metabolites. We have no idea how many of these will turn out to be substrates for FMO3. I know that some of you think that there are other substrates that are causing the odor, and maybe an impaired FMO3 will be found to contribute to this. This will take some time for us to work this out. And I think that is my last slide. So thank you very much, and if anyone has any questions, we'll be happy to try and answer them.
Yeah, that was the last slide here. So thanks very much for that really fascinating presentation. I think there was, so I'm going to read some of the questions from the chat, maybe, to you, and I think one of the first ones that came up while you were talking there was about the choline overload, and if that effect will remain forever. And perhaps that person is going to retype their question too. But if I go up here, yeah, the trimethylamine overload, does it damage permanently? Was their question.
What for precursor overload?
Well, right.
Okay, so for precursor overload, it's if you take away the precursors, then the overload, if you don't provide the body with a lot of choline, then the precursor will be gone. Where it was actually noticed was, I think people with Huntington's chorea, they were given a lot of choline in the hope that this would improve brain function. And what they ended up with was a strong body odor and a lot of production of trimethylaminuria. And once they stopped that therapy, that was fine. The trimethylaminuria production reduced to normal. And that would be true for, if you ate a lot of soya, if you're a vegetarian, you eat a lot of soya, and you're sort of maybe heterozygous for FMO3, you would just maybe have to cut down on the precursors. The problem with trimethylaminuria is, red meat. Doesn't matter, being a vegetarian, soya is full of choline, so a lot of foodstuffs with choline. Yeah, so it's difficult to avoid choline, and it's an essential component of the diet, especially in a developing child.
Okay, great. Yeah, thanks. I think that answered that person's question. And now I see there's a whole bunch of questions from the user Dubia. How do you want to start to address those, the ones that you can address? Do you just want to read them out, the ones that you can comment on?
Okay, I can't see any questions.
Ah, you can't. Okay. Do you have the chat here? Maybe I can make the chat window big. Can you see the chat window now?
Yeah, and it just says hi and welcome. I'm Rob from Europe.
Oh,
I
okay.
can't see any other.
Okay. Look on the bottom. Is there a tab that says everyone?
Oh yes, okay. So I'll just click on that.
Okay. Yeah. Yeah. Okay, so yeah, then if you go to the bottom, or hopefully it'll take you to the bottom, I see some people have typed out those, some questions. We may not have time for all of them, and you may not be the person to answer some of them. So do you just want to have a look there at ones that you would like to comment on, and maybe read them out in comments for everyone?
Let me see. There's quite a lot of chat.
Yeah. So we're all the way at the bottom where Dubia says, "Is there any hope of treatment or cure for chronic hepatitis B-induced TMAU?"
Oh, I'm afraid I couldn't answer that question.
Okay. Yeah, that seems a very specific one.
That would, or I know that it is a rare thing, but it does seem to be permanent, and I don't know if yeah, even when the hepatitis is gone, and we don't understand why.
Okay. Great.
So
There's might be.
Sorry.
Sorry. Go ahead.
I'm sorry. The text is very small here. Can I make it bigger? We have
Yeah. Sorry, I'm not sure how to make it bigger. Let me see.
Sorry, maybe you can.
Here we go. Text size. Yeah. Here, I got text size too. I can get it to go. There we go. Is that better? A little bigger. So the next question was, people with glucose 6-phosphate dehydrogenase deficiency are normally advised to avoid sulfur-based drugs. That is it mean it leads to the inability of metabolize sulfur, and if that is so, can high accumulation of sulfur in the human body lead to body odor? Bit of a confusingly worded question there for me.
That's a different. I'm going to let Ian answer that question. Oh no, he's saying I must answer it. Okay. No, that's an entirely different thing. Glucose 6 dehydrogenase was it glucose 6-phosphate dehydrogenase?
Yeah.
Right.
Okay, now that's different. That is when you're actually perturbing the ability of the cell to balance its what we call its redox potential. It's sort of hydrogen and oxygen potential. It doesn't lead to a buildup of sulfur. It's something quite different. And that's Timothy. Yeah. Okay. I'm just going to let Ian come in here.
Can you hear me?
Yes, thanks, Ian.
You can hear me. Okay. One of the things that glucose 6-phosphate dehydrogenase does, it produces the reduced form of a chemical called glutathione, which is involved in the metabolism of lots of different drugs. So if you have deficiency in this particular enzyme, it's more difficult for you to produce this glutathione chemical, so it will be more difficult for you to metabolize or clear drugs that normally go via this pathway of joining them, what's known as conjugating them with this small glutathione molecule. So I think that's the main problem because 6-phosphate dehydrogenase.
So when a drug has to start its journey out of the cell, it's very simple with the FMOs where I showed you put an oxygen on. But sometimes the drug has to be changed by P450, and then it gets changed and has glutathione put on it. And that glutathione, as Ian said, is the thing that attaches to the drug and helps it on its further journey outside the body to get out of the body. So it's quite a complex process to get some drugs out of the body. It's not just a simple thing like with FMOs, you put an oxygen and it goes through your kidneys and it's out. It can be a lot more complex than that. And so any deficiency in glutathione will influence how you clear the drug, but it doesn't lead to a buildup of sulfur itself in the body. That was the question.
Okay. Yeah. Okay, and the last question from Dubia was, can the TMA enzyme be synthesized and sold commercially like the lactase enzyme sold to lactose intolerant patients?
Well, we can synthesize the enzyme in the laboratory, but any enzyme that is sold to patients. Can you hear me?
Yes, sorry.
Yes. Yes, we can synthesize the protein in the laboratory, but it's the delivery that's the problem. And also anything that would have to be given to a patient has to undergo very strict guidelines. So, and there's a lot of complications in that.
Okay. Next.
In theory, yes, but in practice, it's a lot more difficult.
Okay. Next is from a user named Olu that's saying, is there anything more effective than antibiotics in clearing the bacteria in the colon? Are there cancer treatments that would work?
Well, the problem is you don't want to clear too many bugs from your intestine. So it's, you know, it's a certain type of bacteria that produces the trimethylamine. I can't comment on any cancer treatments that would, I'm sorry, I'm not a clinician, so I couldn't give you any advice on that. Antibiotics are used as a short-term measure, but they're not a long-term treatment. And you need, as far as I know, you have to treat different classes, but I mean, you know, the patients themselves will be much better if they've had the treatment of knowing whether it's effective or not. As far as I know, and from what patients have told me, is yes, it kind of works, but then it's not a long-term solution. And again, taking antibiotics long-term is not a good idea, because those bacteria are needed for health. Generally, the bacteria, because taking bacteria, you'll kill other bacteria as well as the ones that are able to produce the trimethylamine.
Okay, thanks. Next, user J is asking, can benzodiazepine or other drugs and their reactions be used as a sort of a cheap way to check for TMAU? So like a different form of a test.
I wouldn't think that would be a good test. A urine test is, you know, kind of really good for trimethylamine. Taking a drug, yes, you can, but it's a bit more complex. The analysis is a bit more complex. And there might be other factors that influence that particular drug as well, you know, like the metabolism of it. So you come out with, you know, it's just chemically, it's more difficult. The trimethylamine test is really validated, so I'm not sure why you would want to change it. I can't think of a good reason why you would want to change it, but maybe the person who asked the question has a good reason. I don't know if they'd like to share the reason.
Yeah. So yeah, that was the user J. If they want to comment again on that, we can go back to that. Next is from Olu again. Could viral meningitis cause the diminishment or the diminishing of FMO3? Okay, so Jay was asking that question because perhaps they don't have access to tests, to the urine test. I think that's an interesting point. Could you maybe talk about your what you recommend for people to get tested? Because I know that's an issue that comes up a lot.
Yes, that's difficult. So being in the United Kingdom with the National Health Service, the test is available on the National Health Service. But that's probably not the case in most other countries. There are some private laboratories that provide the test, and I think MEBO were trying to set up a test system or a test center to help people. Yes, I don't know if Maria wants to comment on that, or Paul, but there is a direct... Maria, do you want to comment on that?
Well, yes, apparently not too many countries have the TMAU test, and that was MEBO's primary effort, developing a new TMAU source. I contacted some of the original laboratories, and they were not able or willing to do an international shipment, so that's where MEBO stepped in, and then we found the Cleveland Clinic that is willing to participate or create a program with MEBO where we can ship kits to practically anywhere in the world at UPS or FedEx service. And also Nigel Manning in the UK is able to extend it to other European countries. So we have that system set up as well. But one of the questions I wanted to ask you, Dr. Shephard, about the TMAU test is that many people who get the kit seem to have a difficult time putting the choline or taking the choline load. I wish there would be another way we could test for TMAU that would not require such a strong choline load, because of the symptoms then will linger forever. you know.
Yeah,
Do you have any suggestions?
That's a real problem. But if you do have trimethylaminuria, you shouldn't necessarily have to take a choline load. But Nigel Manning would be the person who could really answer that question. But yes, choline does have effect. I mean, the original test was actually done with trimethylamine, so you can imagine how distressing that was.
Right, and I agree with you, because in the case of my son, he refused to do the choline load. He only had 2.5 beers the night before, and even though his TMA level was low and so was the TMAO level, the ratio, TMA and TMAO ratio, was indicated possible primary or genetic TMAU. But then I suppose that without the choline load, you're only really looking for primary, and it really wouldn't measure secondary. Is that correct?
No, I think, well, I would have thought it would measure both. I suppose the problem is if you are severe, you're going to find it. If it's mild to moderate, then maybe you need the choline challenge to confirm that.
Okay.
I kind of feel like, Nigel Manning is the expert on this and would be much better. I mean, he does the test and has always helped us out when people have asked for the test. So, as I said, I'm not a clinician, and nor do I do the urine test. But I think anyone who thinks that they have primary TMAU and doesn't want to do the choline could certainly opt not to have the choline challenge first, because it will certainly show up if you are severely affected without any choline challenge.
Thank you.
Okay, maybe we'll try to answer a few more questions here. So somebody was asking, Oli was asking, could viral meningitis cause the diminishing of FMO3?
I honestly don't know. I don't, unless you've read a report. I haven't read a report on that. Has anybody? I really don't know. That's to do with the brain meningitis. And I don't know if the question's been asked because someone had meningitis and then developed TMAU. Do you mean meningitis or hepatitis?
I think they've written here, yeah, they've written here viral meningitis.
Viral meningitis, yeah. So that's kind of hard to understand how that would cause TMAU, but I'm always ready to, you know, I've never heard of that. I've never heard of that. I don't know if anyone has.
Okay. Next, Adrian is asking, I have secondary TMAU, and they've given some like lab values here. 28 U mole per M mole, TMAO is 0.14. Even when my TMA levels are low and the FMO3 function is high, I can still excrete foul odors. Could the FMO3 function fluctuate between different chemicals?
I mean, the odor might not be due to trimethylamiine. And with secondary TMAU, there are also some indications about kidney function. So if you have a kidney problem, then you could have problems clearing the TMA, and that might cause a buildup. But not being very helpful on this question, I'm afraid. But it, as I
No.
Sorry.
Sorry, keep going.
No, so you know, if the enzyme is defective, yes, it's possible that it could work better with one chemical than another. It just depends on the chemical, and that's true. It sounds like a yeah, sounds like an ambulance going by. Yeah, so I think the question was whether you could have a difference with different chemicals, and that, yes, that's certainly possible. But perhaps the kidney function test might also help.
Okay, so yeah, it seems like, sorry.
No, I was just going to say that the odor might be caused by another chemical that's not trimethylamiine and that we don't even know is a substrate for FMO3. There's so many chemicals and we just don't, you know, don't know. And trimethylamiine is just one of the odor chemicals. You know, there are lots of other odor chemicals. I'm not saying that those are from FMO3, but you know, there are other enzymes. There's also another enzyme, dimethylglycine, that can also produce trimethylamiine.
Okay, the next question is from Olu again. Is there a way of creating an active FMO3 in the lab?
Yes. So when we check to see if a change changes the activity, we create the change on the DNA, but you can go in the reverse way and change the change back into the normal thing and create it active. But that's in, I don't know if the question is, could we take someone's gene and make it active? That we couldn't do. Ethically, we couldn't do that, but we can certainly do it in the laboratory, you know, just as an experiment. I'm not sure if the question was thinking about sort of gene therapy.
Yeah.
Gene therapy. And yeah. Look, enzyme therapy has been used for some disorders. That's true. It's effective for some. But there are problems with any of these therapies, and they're all very, very expensive. So, time will tell. I mean, things will happen. I mean, gene therapy, that, changing someone's genetics, that would be, I think, would not ever be accepted. To change someone's DNA for trimethylaminuria, because I know it's a very distressing disorder, but it's non-life-threatening in the sense of some other disorders. So the ethics of that are very difficult. Other therapies, however, where you can replace things, of course, that is ethically permissible, but will take a lot of research and a lot of money.
Okay, I think that answers. Sorry. I think that answers Jay's question then too about what are the possibilities of designing a supplemental enzyme. So we'll go next to guest BB, who said, how can we become bacterial overgrowth in the intestine? Are there also other microorganisms as bacteria that cause overgrowth, like amoeba or other materials? And can some microorganisms survive in other organs of our body? Sorry, that question is a bit complicatedly written, but does that make any sense to you?
Well, tuberculosis, the bacteria are all over the place, in macrophages and everything, and inside cells. So bacteria do get around the place in the human body. I'm not sure that anyone has correlated trimethylaminuria with bacteria in organs other than in the gut. But maybe nobody's ever looked. Certainly, when you've got a disorder like tuberculosis, that's a huge number of people are affected, and of course we know a lot more about that. So we know that Lyme disease, they go into the cells in the lungs, they go into the brain. You can have very devastating effects when the bacteria end up in the wrong places. And I'm not aware that that's the case for trimethylaminuria. And
okay, next is from. Sorry.
No, I was just going to say we're not really sure what causes the bacterial overgrowth. We do know, for example, in obese people, their bacteria are different to someone who's thin. So but there are not enough studies on trimethylaminuria patients to know how their bacteria might be different to someone who does not have trimethylaminuria. But some secondary.
Okay. Sorry.
I was just going to say that some secondary trimethylaminuria can be caused by changes in the bacterial gut, but actually we don't know enough about what changes them, or what the cause is. We do know that people who've had some intestinal surgery then go on to develop changes in their bacteria that can cause secondary TMAU. That's one class of person, people who have complained, but not everybody who has intestinal surgery, of course, because it's very common. And maybe that's also because of genetics as well. It's very complex, this interplay between the gut and the rest of the body. And we're really only starting to study it, let alone understand it. I mean, not me in general, but the whole scientific community.
Okay, next Olu is wondering, are painkillers like Nurofen using the FMO3 substrate?
I don't think so, no. I don't think there's any studies that show that ibuprofen is a substrate for FMO3. FMO3 doesn't.
Okay.
I was just going to say FMO3 has quite a number of drug substrates, but it's the actual number of drug substrates is much smaller than, for example, FMO1, which is another FMO protein that, or cytochromes P450. So most of the chemicals that are metabolized by an FMO are also metabolized by P450. So having a little FMO may not actually influence the metabolism of the drug that much, because you've got your P450s backing you up. I suppose where you would get into a problem is if your one of your P450s was also had a problem, and then the two would both not be doing the job properly, and then you might have a problem. But again, that would be dependent on the drug. So I just don't want people to worry about drug metabolism. Obviously, if you've had an adverse effect, then your doctor would be able to advise you on that. But it is complex. It's not just sometimes it's not just one gene, one drug. We have all this multiple drugs that are acting to help us clear drugs. And if you do have a problem with one drug, then your clinician should be able to advise you to take another drug of the same type that's not metabolized by, for example, FMO3, if that proved to be the case.
Okay, another question from guest BB. If you did a blood test and it's negative, is it very sure that you have TMAU2, so secondary TMAU, than if your urine is positive for TMAU?
Okay, so if you are diagnosed with trimethylaminuria from a urine test, but your genetic test is negative, now then you may not have primary TMAU, but you might have. And the reason I say that is because we don't know all the mutations that cause trimethylaminuria. And if it's a mutation that's, the gene is very big. So the protein, I said, was 532 amino acids, so that's almost about 1,600 bases of 1,600 A, T, C's, and G's. The gene itself is over 30,000 bases. So we know most about the changes in the region that codes for the protein, but a gene is much more complex than that. And so if there's a mutation somewhere else, a mutation that causes a low amount of protein to produce, we might not know about that. There might be a mutation in what we call an intron that's part of the gene that is also causing a problem. So a negative DNA test is only as good as the information that we have to interpret that genetic information. It is, the person, you could still have primary TMAU. So, but it could be secondary TMAU. I think if you see the problem, unless you have your whole gene sequence, and even then you'll have so much information that it's hard to interpret without doing a proper experiment that will define whether that change influences the amount of protein produced or its activity. So the simple ones that we can define very easily are the ones that change something in the protein itself. And that we can test very easily in the laboratory. Testing for mutations in other parts of the gene that change the protein's amount or the protein are really quite difficult. You'd have to, we would have to test it. We have to have a functional test to show that. I don't know. Maybe you don't appreciate how difficult this can be. You would be looking for a needle in a haystack, if I can put it that way. But if you had a whole family that had the same mutation and it's segregated with the disorder, then you could pin that mutation down to something. So if a person has TMAU and has no family members, or that also have the disorder, then maybe it is secondary TMAU.
Okay, next, Jay is wondering, could long-term use of antibiotics have caused TMAU?
Depends which type of bugs that would be killed by the bacteria. So was the question that the actual antibiotic itself would cause TMAU?
The question was long-term use of
Long-term.
antibiotics causing TMAU.
Okay, in theory that might be possible, because depending on the antibiotic used, different antibiotics would target different types of bacteria. So if you were, say, selectively killing off the bacteria that would not produce much TMA, that would mean that other bacteria that produced lots of TMA might increase in number. So in that sense, if you were selectively killing off certain types of bacteria, you might have a gut population of bacteria that would end up producing more TMA and hence causing secondary trimethylaminuria. But that's not really well understood. But in theory, yes, it could be possible, because you would be, if you were changing the population of bacteria in the intestine in that way, you might end up producing more TMA.
Okay, thanks. The next question, I lost my place here, is can FMO3 not be used like insulin?
Sorry, can it, you, in other words, like an injection? Is that what?
Yes, I think that's what they mean by.
In theory, insulin works in the muscle. And if you consider how much time and research has gone into insulin, in theory, of course, this would be possible. It's getting it to work in practice that would be difficult. Well, it would be challenging. I wouldn't say it's impossible. It would be challenging. And that's just simply because of the nature of those types of therapies. They're all possible, but they're challenging to get it to work properly. So insulin, you know, it's been years and years and years that we've known about insulin, and it's been refined, and you can inject it, and it's taken up into the muscles where it works. So it's slightly different. But in theory, yes, in time, maybe, perhaps.
Well, one of the difficulties here is that with the insulin case, the insulin's being injected usually intramuscular, so it's into the muscles, and that's where insulin has most of its action. It will increase the uptake of glucose into the muscles and hence clear the glucose from the blood and therefore help treat the diabetes. But when you think about it, FMO3 is working mainly in the liver, so it's very much more difficult to, say, target an FMO3 to the liver if you were just to inject it intramuscularly. So it's a more difficult problem than intramuscular injections of insulin.
Okay, thanks. Next from guest BB, can my kids develop
If someone has secondary TMAU and it's not genetic, there isn't any reason why the children should develop this. Only if there's some genetic component that they've inherited. But I don't know of cases of inherited secondary TMAU. because it's basically not inherited, if I haven't put that very well. So the primary
Okay, next is... Sorry, I tend to just start talking at the worst time there. Next is from Janet. Do you think Hy pylori, HPYLORI,
I don't think that it is. The particular bacteria that produce TMAU are a class of bacteria that are called methylotrophs. And they are able to utilize through a very unusual reaction the trimethylamine as a nutrient. I don't think that that the species you've just mentioned has been implicated in TMAU. At least I haven't heard of it. I don't know if Ian has. No, he's shaking his head. No. But that doesn't mean it isn't. It just means we don't know that it is. We don't know that.
Okay, next is from Marcella Davis. Do you know, or can you tell us specifically, what type or types of bacteria are involved in creating the environment, bacterial overgrowth, conducive?
Well, it's this class of bacteria that are called methylotrophs. Okay, there's more than one type, but they fall into this category called methylotrophs. And as I said, they just have this property that they, actually what they do is, we always show it simply as choline going to trimethylamine, but it's choline which goes to betaine. They then have an enzyme that can then create the little trimethylamine molecule, and then they do another very complex reaction which gives them a carbon and nitrogen source. But they're a very particular class of bacteria. And the problem is that once they produce trimethylamine, they are creating a carbon and nitrogen source for themselves, and so they grow even better. So they kind of are creating their own food cycle, if I can put it, from the trimethylamine. And so then, and because they're creating this sort of nice nutritional basis for themselves, they can outgrow the other bacteria. So there are lots and lots of different bacteria in the gut. We'll never know how many different types there are, but it's these methylotrophs. But it's the balance. It's the balance between the different types of bacteria that are important. So if the methylotrophs increase in number, they will produce more trimethylamine. And as I said, once it starts, you know, they perpetuate themselves because they are creating this very nutritious environment for themselves.
Okay, next is from Olu. Could viral meningitis cause the enzyme to be destroyed?
Well, meningitis affects the brain. So we do have evidence that there's FMO3 in the brain, but it's not, you know, the primary, I don't know why it would cause it to be destroyed. We have no evidence, I have no evidence for it, and I don't know of any evidence for that. I wonder if the person, maybe they don't want to tell us why they're asking that question, or whether they know someone who's had meningitis who then has developed symptoms of TMAU. But I don't know of any published report. That doesn't mean there isn't one, of course, but I don't know of one. Because the FMO3 is very, very predominant in the liver, with a small amount in the particular section of the brain. It's only a very small amount in the brain. It's an enzyme that really is confined to the liver.
Okay. Where are we now? Okay. Nemo's wondering, I've had 2 TMAU urine tests. Both came back negative for TMAU. I had a choline overload for both tests. What should I do next in terms of diagnosis? How should I approach my GP about getting
It may be some other body odor and not TMAU. Because if, can you hear me? Okay. If the test is negative after a choline challenge, and twice, it's possibly not a defective FMO3 that's causing the problem. The other gene, there is another gene that's been found to increase trimethylamine, but there are other body malodor disorders, you know, some that we don't know the basis of. So the person may not have trimethylaminuria. It may be another disorder. I just think it's unlikely if the test has been negative. If the test has been done, they could approach their GP. I don't know who's done the test, but if the test has been done at an accredited centre, such as the one at Sheffield in the UK, and it's negative, then it does seem that the test, if it's been done on 2 different occasions, it does seem that that would rule out TMAU. So the source of the problem must be something else. And there are other, as I said, there are other disorders that can cause body odor. And sort of ammoniacal smells. Citrullinemia is one, but that's quite how can I say, that's something that is much more, I don't want to diminish TMAU as a severe disorder, but citrullinemia is...
Okay, next Daisy is asking, what about fecal transplant to repopulate the gut with friendly bacteria?
It can cause mental problems and things like that, but it also has a similar kind of ammoniacal odor when the child is going through an episode. Well, they've tried this where I think they took the bacteria from a thin person and did a transplant into a fat person, because the bacteria are really quite different, or the classes of bacteria are quite different. I think it worked temporarily, and then the bacteria reverted back to what been the fatter phenotype. But yes, in the future, that's not...
Okay,
you know,
next.
People are thinking about these things in terms of other disorders, particularly for obesity. And, you know, that's really where you see the very striking differences in the distribution of the different classes of bacteria between obese people and people of normal weight. No, I think it might, I can't, I've never done a fecal transplant, so I can't, I only have looked at mouse feces rather than human feces, I'm afraid. But I think people are thinking that way for other disorders. And if it works, you know, for something like diabetes, then, you know, the knock-on effects are, well, okay, then it knocks onto rare disorders. But you know, the money always goes for the bigger disorders first, and then knocks onto the rarer disorders later. But to manipulate the gut most effectively is to eat a balanced diet. That's the easiest way, is to eat a balanced diet. Or perhaps some sort of probiotic or something to help the bacteria to sort of be at its normal spectrum. In other words, the normal distribution of the different families of bacteria in the gut.
Okay, next.
There's a problem. Yeah, so if someone has an imbalance, then eating a balanced diet, you know, or probiotics might help them. I mean, I'm not saying this as a clinician. This is just an opinion. I'm not saying it will. But a balanced diet is always the best way to go. You mean in a urine test? Yes, a urine test should show whether you have secondary TMAU. You will have it will show if you've got TMAU. It may not show that you've got secondary TMAU. And then a genetic test would then determine whether it was primary or secondary. But the first test is always the urine test. That's the least invasive, except of course the choline challenge isn't pleasant. And then there's the genetic test.
Okay, next, Christian is wondering, can certain...
That as well, that people have to consider. Some people want to know and some people would prefer not to. I think that's a personal choice. But I would always recommend urine first and then genetics afterwards. I don't think anyone's done a study that is large enough and sufficient enough. Oh, riboflavin, yeah, that's not an amino acid, but you know, some different people find different things work for them, and if it works for them, that's great. But you know, a big study that's controlled, that really says this works or that doesn't work, have not really been carried out. But riboflavin has helped some people. As has, if you've got some FMO3 activity and it's not completely knocked out, then riboflavin supplements seem to help some people. Because one of I mentioned that, you know, the name is flavin containing monooxygenase. So if you're deficient in the flavin and you can boost up the amount of flavin, then the chances of the little molecule binding to the protein
Okay, next observer is asking, do you think people with FMO3 deficiency may be prone to other vague or acute health disorders other than malodor? For example,
should be followed. But certainly some individuals have found riboflavin to help. Oh gosh, I mean, you know, the people who are on this webinar can probably answer that better. I've not, people haven't really mentioned that.
Okay, next Maria is wondering, what are the differences amongst adverse drug reactions?
Or with fatigue, but you know, I stand corrected by the people who actually have the disorder to correct me there. But there's no documented evidence for that. You know, patients might have those symptoms, but there's no documented evidence. Okay, so an adverse drug reaction would be when you take a chemical and the concentration of that chemical rises to an amount that to an amount of the drug that would cause a very bad effect. So, for example, if you had high blood pressure and you were taking a tablet for high blood pressure, but you couldn't clear the drug, then your blood pressure would probably become too low and you'd collapse. So that would be an adverse drug reaction. You're reacting, an adverse reaction to that particular drug because you can't clear that particular drug and its concentration is rising. I think the next part of the question was about allergy. Allergy is something different. Allergy is when the body responds to a chemical. So, for example, if you have hay fever and you react to a pollen, your body, what causes the hay fever is your body responds to these, for example, something in the pollen, and it produces a chemical called histamine. Then the histamines rush to the site of where the irritation is, and that's why when we take antihistamine drugs, they reduce the amount of histamine and they reduce the amount of the allergic reaction. So they're quite different. So the adverse drug effect is in response to this therapeutic drug that you are taking and you can't clear, and the drug amount is rising. The allergy is when your body, your immune response in your body is responding to something, and it's oversensitive. So allergic reactions involve an immune response. An adverse drug reaction is a response to the drug. Now, I'm sorry, the third part of the question was about chemical sensitivities. Yes. All right, so there are some people who are, but in some ways the chemical sensitivity is also an allergic reaction. The body is responding. Well, actually,
so
a chemical
Dr.
sensitivity,
Shepherd, in the case of having a reaction to a general anesthesia where you're really having like vomiting, nausea for hours, would that fall more under adverse drug reaction or allergies or possibly even both?
to that chemical. So that's a little bit more complex. So it could be a mixture of both in that case.
Mm-hmm.
It could be possibly both, but also some people respond badly to anesthetics because of other mutations in other genes, okay, that are involved in anesthesia. So it's not
like coming out and yes, like coming out in recovery, like during the surgery, a 3-hour surgery, everything was fine, but then coming out of the surgery and recovering,
to the chemical.
then there was a 4-hour seizure, vomiting, nausea.
If it's a reaction to the drug would have to build up in the system for the response to occur. It would be a slower response.
Okay.
Does that make sense?
Okay, next, Daisy. Sorry, keep going.
Yeah.
Okay. Next, Daisy is wondering, where do you see the research?
Yeah, well, that could very well be an adverse drug reaction. And, something that... No, carry on. Oh, if only. I think that, you know, of course we would love to be able to tell you that we had a therapy that's going to make everything right, and of course we'd, you know, people are trying very hard to write proposals, to get money, to do the research. I can't say any country's at the forefront at the moment. The UK, of course, has, the UK and the States and Japan are probably the three that are where the most work is being done. But getting money for scientific research is always difficult. All I can say to you is we keep trying, and I hope that one day I'll be able to say to you we've been successful, and this is what we're going to do to try and help. But it's you know, getting scientific research is incredibly expensive, and of course, you know, the economic situation of the world is not in the greatest shape. So it was much easier to get money to define that FMO3 and TMAU urea was a genetic disorder associated with FMO3, but to move on from there has proved a little bit more difficult, actually a lot more difficult. So I'm sorry that I can't give you more optimistic notes on that, but there may be other groups out there you know, that I don't know about that are you know, processing along the way. But if they are, they haven't published it. I would also say Australia is also, there's a very good group in Australia also working on TMA. There's individuals all over the world. You know, there's not many of us, and we do know, most of us know each other.
Rob, are you there?
So if there is someone out there...
Sorry, I had my microphone. No, I'm here. Sorry, I had my microphone muted, so I was talking to myself there for a moment. Sorry, Observer is wondering if someone wanted to reduce their FMO3 substrate input, is there any ways they could do this that would make a difference? For example,
Well, I don't know about filtering tap water unless you live in an area where the water is recycled a lot and has got, like London, it's recycled 7 times apparently. But you know, that would just depend on the chemicals in the water. A diet that is low in choline, that, but you have to be careful here because, you know, if it's children, what you would want to avoid are, or at least limit, are things like eggs and liver, kidney, Brussels sprouts. You might think that's wonderful. That's something you probably don't like, and you would like, and broccoli and cabbage and cauliflower. Peanuts, anything containing a fish oil supplement you probably should avoid. Health food supplements that contain fish oil. We've talked about choline, but lecithin is used as a preservative, you know, like in salamis and things, and lecithin also is acted on by the gut bacteria to produce trimethylamiine. Seafood, fish that live in the sea, marine fish, you should avoid those because, or cut that down. River fish should be fine. The reason for that is fish that live in the sea have to cope with very high concentrations of salt, and one of the mechanisms they use to help to stop their proteins changing shape when the salt concentration of the sea changes is actually trimethylamine N oxide. So seawater fish have a lot of trimethylamine N oxide. And so if you eat sea fish, marine fish, then the bacteria in your gut actually take the oxygen off the trimethylamine, of the oxide to produce trimethylamine. So that's a very rich source of trimethylamine. But river fish don't have to worry about salinity. So they don't have a lot of trimethylamine in oxide like marine fish do. I don't know what the person meant when he said organic food. He meant organic food is kind of an emotive issue because all food is organic. Organic means that it contains carbon. All food is organic. It's just whether you want to have things that are grown without being sprayed or something like that. But there's a lot of natural pesticides, in plants. Plants produce their own pesticides to protect themselves. So I think you just need to look at the labels on your food, on food, any processed food. Look at the label. If it's got lecithin on it, don't buy it, don't eat it. As I said, fish oil supplements, people think these are really healthy, but if you've got TMAU, they could certainly make things worse because they might have a lot of carryover of TMA and oxide or TMA. And different people find different things work for them. I mean, you're the experts. I'm really not on what you found works best for you. But again, it's a balanced diet. It's avoiding the preservatives that can give rise to trimethylamine, avoiding soya products because that's high in choline, peanuts high in choline, actually peas and beans. I'm not sure if I've mentioned all those.
Okay. Are there any last comments that you would? I know we have a few more questions and written here, and I'm sure people could write questions all night. But I just want to not take up any more of your time here. So maybe now's a good time to maybe wrap up. Are there any final comments that you want to tell the 20 or so of us on this webinar?
No, not really. I appreciate you would love me to to say that we've got some magic or wonderful, substrate that you could take and this is going to make the symptoms go away, but we're just not at that stage yet, and I just really hope that we will be within, a while, and to have better news to report to you. I can only thank all of you for attending. I'm sorry that we couldn't be more positive with some of the questions. I'll just
yes, and
I would like to say something, if I may,
and you're listening to do have the urine test, and to those who've had a test,
Dr. Shephard and Phillips, I'd like to thank you very much on behalf of the whole international community, not only for this presentation, this webinar, but also for the dedication that you have proven, that you have shown us to educate us with all your PowerPoint presentations, and of course this very informative website. You seem to have seemingly endless patience in answering our questions, and not only now but throughout the years. And we are indebted to you for this kindness, and your compassion is palpable. We can really see how you really care for us, especially when you do all this without us having to pay you, because we don't have the money to pay you. I wish we had a fortune to give you for research. but maybe someday we will, I hope. But for now, all we could give you is to express our gratitude, our very profound gratitude for everything you have done for us. Thank you.
Okay, yeah, I think Maria said it perfectly. Thanks very much, Dr. Shephard and Dr. Phillips. And my final comment is just I'll share in the chat window here the link to the TMAU community on Rare Connect, where hopefully we can post this webinar tomorrow. And just want to encourage you to keep discussing with each other, and please feel free to use Rare Connect as a place to discuss and share your experiences, so that you can all benefit and learn from each other. I think we had a great discussion tonight with some researchers, but I think also patients connecting to other patients can result in some good knowledge being shared. So yeah, please consider that as well. That's about all. Have a good night or a good day, everyone, and we hope to have more webinars like this in the future. And I think this was a really great start, so thank you.
Thank you.